me. I'm the chemistry RPM for proteins. And I'm just going to start off by saying that research is very, very frustrating and can often be correlated. That's true for every discipline, for every major, but I really think that that's true for biology. I'm a biologist. And for biologists, your research is really only as good as whatever model organism you're using, whatever cells or mice or turtles or zebrafinches or whatever. If they're not working properly, if they don't want to cooperate with you, if they're giving results that just don't make sense, which happens all the time, it's difficult. But it's also true that there are times when you get results that are completely contradictory to what you expected, and those turn out to actually be really interesting and even to be sort of mini breakthroughs in a sense. And that's not something I realized until I started doing research on my own. That was 2 summers ago. I was working in a lab that studied melanoma, a form of skin cancer, over on the medical campus in the Central West End. And essentially this lab was studying how melanoma arises, how it can be virulent, how it's bad for you, and also how it can be treated. And originally my summer project was going to attack melanoma from the how it arises angle and how it becomes bad for you. But because of this twist that it took a few weeks into the summer, it actually became more of an investigation into how it can be treated. And I say that because even more specifically, I was focusing on a protein called Pyk2, also called Pyk2, which my mentor and I originally hypothesized would basically help cancer cells to survive, help tumors to proliferate. And the reason why we thought this was because Pyk2 is really structurally similar to a separate protein called FAC. And FAC is known to promote cancer cell proliferation. And because in biology, we always say that structure and function are really closely related. So if 2 molecules have similar structures, then it's sort of reasonable to conclude that they also sort of do the same thing. But when we tested this, that turned out actually not to be true. We saw that Pyk2 actually killed off cancer cells, not what we expected. And by finding this out, or in order to find this out, we basically devised a really simple experiment. We took about 40 plates of melanoma cells, and we divided them into 2 sets, so 20 cells each. And we injected half of those plates, 20 plates, with really high concentrations of Pyk2 protein. And we took the other 20 plates, and we injected them with equally high concentrations of GFP, which we're basically using as an experimental control. And essentially the purpose of this GFP was to provide a sort of basis for comparison, so that if something really, really interesting or really unusual happened in the Pyk2 cells, then we could turn to the GFP cells and go, well, that didn't happen there. So whatever happened was unique to the Pyk2 injections. It's not just some universal effect that you get from stuffing cells with any protein whatsoever. And so after we finished the injections, we took our plates. We allowed them to incubate under fairly warm temperatures for about 48 hours, which actually worked out really well. That was over the 4th of July. And afterwards, we went into lab, my mentor and I went into lab fully expecting to see the Pyk2 cells alive and thriving and proliferating, because that was what we thought would happen based on FAC. But that was not what we saw. And if you turn to the pictures at the bottom of your handout, you can see that the GFP cells looked fairly normal, for the most part, not perfect. But in general, they pretty much covered the entire plate, which meant that they had grown, they had multiplied, they were operating fairly normally. And if you turn to the Pyk2 picture, you'll see that actually looks quite different, because those cells look wrinkled. They don't look as well formed. They're not covering the whole plate. There were a lot fewer of them, which was interesting, because they actually looked as if they were dying. And so when my mentor saw this, she immediately said that could possibly be apoptosis. These Pyk2 cells might be undergoing this special form of cell suicide, which is what apoptosis is. And that was really interesting, because actually apoptosis is what goes wrong with cancer cells. And the reason for that is because essentially all the cells that we have sort of a finite predetermined lifespan. And usually, if they're functioning normally, once those cells reach the end of their lifespans, they'll actually sort of kill themselves. They'll commit suicide in a very organized, regulated process. And so they'll die, and they'll make room for new cells of the same type to come along and to sort of take over what they were doing before. And that's normal cells. That's not what happens in cancerous cells. Basically what happens in cancerous cells is that something goes wrong with the apoptotic mechanisms. And so those cells don't kill themselves when they're supposed to. They sort of stick around even after they've grown really old. And so when new cells are coming along and crowding in, you sort of get these masses of cells forming tumors, which are not good for you because they're generally cancerous and they're malignant. So that was why it would have been interesting to look at this target from the apoptosis angle. But before we did that, we really had to make sure that this was what we saw was real, that it wasn't just something that happened at one time and was due to a fluke or something. And so we did that same experiment again with the same cells. We did it again with an entirely different line of melanoma cells. And both times we saw pretty much the exact same thing, where the GFP cells looked healthy, they looked normal, they were proliferating, and the Pyk2 cells looked not good in general. They were dying and they weren't sticking properly to the plate or multiplying properly. But what was really interesting was that when we tried the same experiment again on normal cells that weren't cancerous, we didn't see this effect happening. In that case, the GFP cells looked good, and the Pyk2 cells looked just as good. So everybody was happy. And why that's interesting is because that shows that this sort of killing effect that Pyk2 seems to be having is probably unique to cancerous cells, because it's not happening in these normal cells. And so if somehow this were to be incorporated into some sort of a treatment for skin cancer, say 30 or 40 or 50 years down the road, then it will be a really, really specific treatment, because Pyk2 specifically targets cancerous cells. It does not adversely affect normal cells, at least to the extent that we tested it. And remember also that this was surprising, because my mentor and I started off thinking that Pyk2 was not going to do what we thought what we actually observed. We thought that it would do the opposite in promoting cancer cell survival and proliferation. And that's because it's similar to that other protein, FAC, that also does exactly that, promoting cancer cell survival. And so, of course, we were thinking about why we saw what we did. And one thing that we came up with was the fact that even though Pyk2 and FAC are fairly similar, they aren't 100% identical. There are still differences, otherwise they would be the same molecule. And it really could be that those differences are key to understanding why Pyk2 sort of functions so differently. One hypothesis that my mentor had was that Pyk2 actually sort of binds and sequesters certain proteins that are needed for survival from FAC. And so essentially what's going on is that in the cells that are getting Pyk2 protein, under normal circumstances, FAC will be helping those cancer cells to sort of survive and proliferate. When Pyk2's there, it sort of hides the tools that FAC needs from FAC. So FAC doesn't have what it needs to do its job. And so if FAC can't do its job, then all sort of goes to whack, and the cells start to die off and to undergo apoptosis. So basically what happened was that we started off the summer with a hypothesis. We thought that Pyk2 would help these cancer cells to proliferate and survive and form tumors, and that when we tested it, and it turned out to be wrong. But we got something really interesting as a result, and we had a lot of fun. We learned a lot along the way. Questions?